Welcome to this year's Florida Lake Watch Regional Meetings. We would love to visit with you in person as we have for all the years, but until this COVID situation settles down, we're going to have to rely on electronic communications. Today I'm going to talk about how your Lake Watch data are used. I often get asked, why should I continue to sample? How are the data being used? What value are they? So today we're going to talk a little bit about the last couple of years of how we've published data, how we use it, how we make it available for everybody. First though, I'm going to talk a little bit about the history. Many of the volunteers are new volunteers. It's always good to recap the history of why we do what we do and what our goals are. So I'll talk a little bit about that and then go over some of the research projects and extension projects that we've accomplished over the last 2 years. Our mission statement, of course, is we want to work with citizen scientists for the purpose of monitoring aquatic ecosystems and provide research quality data. That's a key. We also want to educate the public on how to be good stewards. That's part of the reason we do these regional meetings. And most of you are aware, over the course of the 30 years we've been around, we've run probably 45 different graduate students. We teach an undergrad class on water chemistry. So the teaching aspect is also a very big one for underneath the Lake Watch umbrella. All of this is done under the umbrella for some type of lake management or aquatic ecosystem management. So the backbone of our program is underlined by this question. There's several thousand lakes in the state of Florida. Go ahead and think a little bit and put a name on the one you think is most important. We think the most important one is My Lake. That's what brings our volunteers every month out to sample. They love their resource. They want to be good stakeholders. We hope the data that we collect and gather and the information we put together helps everybody understand their lake and help them become better stewards of the systems. Of course, Confucius is tagged with this statement, but it's really a librarian that used to work for him. Just a little history lesson. Journey of 1,000 miles begins with a single step. The single step for Lake Watch began in August of 1986 when stakeholders at Lake Santa Fe, they were not getting a lot of information that they really wanted to be good stakeholders. So they ended up at the University of Florida, talked to Dr. Canfield and myself, and we gave them bottles, said if you go ahead and sample for a year, we'll gather up and talk about what we know about the lake and give you the best information we can about it. Of course, Dr. Canfield and I were busy at the time doing a lot of research on hydrilla and fish populations and how to manage that plant, other exotics like hyacinths. So we had a pretty big research interest and we didn't have the time to go out and take samples ourselves. At the end of the year, the Santa Fe people had collected monthly samples. We analyzed them for nutrients and chlorophyll. Talked a little bit about the trophic state, other things about the lake. They were very pleased, asked if they could continue sampling. We said, yeah. And then a neighbor lake, Lake Alto and Lake Hampton and Lake Broward, all heard about this and they wanted some of that action. So before long, we were sampling a couple hundred lakes. And this was really good for the research aspect. The one question Dr. Canfield and I pondered is how the geology, which is very diverse in Florida, impacts the water chemistry of lakes within those geologic areas. The only way to answer that question is to sample as many lakes across the state as possible. And so all of a sudden, we realized we had a free workforce to go out. We were helping them and they were helping us, so it worked out great. The interest grew so much that in 1993, the Florida legislature actually made a statute creating Florida Lake Watch as Florida's citizen science program at the University of Florida. With that came a little bit of money, and you can see the rest is history. We rapidly increased just by lack of funding or inflation. We've kind of tapered off, but we're still managing to maintain about 580 lakes, 130 coastal sites, 125 river sites, and 5 springs. This fluctuates annually as people retire and we train new volunteers on new systems, but we try to maintain, this is about where our budget can hold. You can see we're in 57 counties scattered all over the state of Florida. That has given us a wealth of information for us to be able to answer those research questions about geology and other aspects, which I'll talk about a little bit later. On the way, one of the big stumbling blocks that we had was that people, agencies, DEP, water management districts, some county governments were skeptical that whether or not volunteers could collect research quality data. So early on, we had done a comparison with our biologists and the volunteers and came up with similar findings that there was no difference between the professional and the volunteers. However, a lot of people were uncomfortable with that because all of our data were run through the same laboratory, which was our LakeWatch laboratory. So around 2010, I went to Tallahassee and we talked to Florida Department of Environmental Protection and lined up a research project where we sent their scientists out with our volunteers, and I got out of the way. They ran their samples through their high quality assurance, quality control program, and we ran our samples through our program. And as you can see, for phosphorus, nitrogen, chlorophyll, water clarity, there was no difference between the data. So finally we put that to rest, and well-trained volunteers, we're very certain can collect research quality data, which is one of our goals in our mission. With that, we finally did a laboratory audit where they came down to our laboratory, watched us run samples, watched how we recorded all the information, gave us lots of suggestions that we weren't quite meeting standards they need to be able to load their data up in their storage and retrieval system. Finally, we agreed to all the changes, and in January this year, we've agreed on a SOP, standard operating procedures, and they signed off with the Deputy Director of the Division of Environmental Assessment and Restoration. With that, we've got our new standard operating procedures. We had to completely change our laboratory spreadsheets to add additional data that was required for upload to storage and retrieval stuff or databases. With that too came a change in how we need the volunteers to record data on their field sheets, which is very important so that we can gather all the data required by our agreement with Florida Department of Environmental Protection. So here we need to make sure we have sampling times. Every single station has an individual time that's required to be able to load it up. We need a filtering date for our chlorophylls, and we need a filtering time. So these are all things that our volunteers now are going to have to add to the new field sheets. If you don't see these on your field sheet, then you're probably using an old one. Call a regional coordinator or look on our website, and our new website, which please take a look at. We're very proud of it, and download the new sheets. So with all of that, we're able now to load our data into the new system called Watershed Information Network, or WIN. That data is also transferred to the US EPA's storage and retrieval system called WQX. And once it's in there, anybody in the planet can get at it. You can go and try to download yourself. It's a little cumbersome. Or you can go to our website, which we encourage you to do. We have a spreadsheet ready to download. It has annual means since the beginning of our program in 1986. You can sort by county. You can find individual lake data. Take a look. If you need the absolute raw data, you can try the Florida Lake Atlas, where we send our data and it's uploaded there, or you can actually call us, and if you need the absolute raw data by individual station, it's too big to put on the website, so we can actually get it to you if you put in a request. So now what do we do with all that data once we have it? Well, that's what I'm going to talk about here. We have, in the last couple of years, probably 10 different publications, internationally peer reviewed. I'm going to talk about a few of them today, just to give you the breadth of some of the things that we do and the reach of our data across the planet. As I mentioned, this is the face of our website. Of course, one of the major things we want of our data is we want to get it back to the volunteers so that they can use it. They can look at it, try to understand how their lake's functioning over time. Each individual county has several lakes per county, sometimes more if the county has lots of lakes, but you can download by county and then sift through until you find your individual lake. Same with estuaries, dune lakes, and rivers. Historically, we collected plants. We've left those reports up there. They're a little dated, but they still have valuable information about what type of plants grow where. The file I was talking about for annual means has headers. That's the title at the top of each column with units. We have the definitions there so you can understand exactly what each one of the headers is, and then the data file can be downloaded here. There's the Florida Lake Atlas, and if you want to get in and try to use WIN, there's the hot link for the Florida Department of Environmental Protection. So getting on with the data, I know there's a lot of big databases out there because I've worked with them for the last almost 40 years. And one of my intentions or research interests has always been to put all these databases together and utilize them to address lake management issues. With that said, I put in a little proposal to Florida Fish and Wildlife Conservation Commission, and I got it to put these databases together. One for sure was the long-term fish database that Lake Watch has been involved with since 1999. In fact, we helped start that process. And during the time when I got the money, as you all aware, there was a big cry for of concern about herbicide treatments across the state and what they were doing to the lake and the fish. It got so loud in social media and stuff that in January of 2019, the Florida Fish and Wildlife Conservation Commission, who's in charge of permitting all herbicides treatments, put a pause on all of those and went around the state for a couple of months before they started again with the treatments to listen to stakeholders and try to understand what their concerns and how best the concerns could be addressed. So out of those listening sessions, two of the main statements, and I kept hearing this from different volunteers also, was that the statewide herbicide treatments are toxic to fish and are hurting the largemouth bass populations, which is one of the main draws to Florida for tourism and anglers. The second one is that the use of glyphosate, it's the active ingredient in Roundup, and it has phosphorus in it. It's used to control water hyacinth and other plants. So people were making the statement that just by spraying, you were adding so much phosphorus that you were causing these harmful algal blooms that everyone heard about last year and the last couple of years. So I put these databases together to address these two questions, utilizing our Lake Watch water chemistry and our plant data and other aspects of our program. And you can see here, I had the actual records for the last 10 years of all the herbicide applications in the state because they have to be permitted. And I divided lakes into low herbicide, medium, and high herbicide lakes. This is a log schedule or scale. So you can see that it's a big difference between high and low herbicide treatment lakes. The high herbicide lakes have almost 3,000 grams per hectare and the low only 26. And then we had fish population data in all of those divisions. So we compared low herbicide lakes with high herbicide lakes and found no difference. And this is young of year largemouth bass. They're probably one of the more sensitive aspects. But we looked at adults, trophies. We looked at all kinds of aspects of fish populations. I don't have time to go through each one because I want to cover several different projects. But the conclusion with all the amount of analyses we did is there's no evidence that herbicides are negatively impacting the largemouth bass populations, addressing that first concern. The second one, again, I had the herbicide information, and here's Orange Lake, but we did this analysis on almost 100 lakes in the state because we have available data once we put all the lakes long-term databases together. We knew how much glyphosate was used. After that, we can figure out how much phosphorus, because of the chemistry of the molecule, is in glyphosate. So we know how much was treated. We know the surface area from our Lake Watch surveys, mean depth, same. With that, we could calculate the volume. By dividing the amount of phosphorus by the volume, we can come up with a herbicide concentration of phosphorus that was applied to the lake if we look at the whole lake, which is less than 0.2 micrograms per liter, very, very low. This is the actual concentration in the lake, and that's been 20 years of Lake Watch data, so it's 122. So you can see that the percentage of the actual phosphorus in the lake that could be contributed by phosphorus is very, very low. The conclusion of this analysis is that spraying aquatic plants with glyphosate is so small amount of phosphorus that it could not be the cause of these large algal blooms. So this is how I envisioned utilizing all these long-term databases. Now that we have them and continue collecting them, we will be able to use these things to address this type of management concern. At the same time we were doing that project, I was involved with others from the Department of Fisheries and Aquatic Sciences to develop a Lake Istapoga habitat management plan. How are they going to manage the plants in Lake Istapoga? We were dealing with citizens. A couple hundred would come to some of the meetings, and one of their main concerns, hydrilla had disappeared from the system, and they were impacting the largemouth bass population because of lack of habitat within the lake. Their concern was that Istapoga had been treated for the last 30 years for water hyacinth and hydrilla, and they thought the sediments were toxic. Legacy herbicides in the sediments were stopping all submerged plants from growing because they're very few. We went out and collected sediments from Istapoga. We did from Lake Tohopelaga, which at the same time had similar herbicide treatments, but hydrilla and submerged plants were growing extremely well. And we also had controls to compare this. We planted tomato seeds, very sensitive plants, and we did actual hydrilla tubers because that was one of the main concerns in Istapoga. The germination rates for all of these were almost identical. We could find no difference in Istapoga and Toho or the controls. The tomatoes grew very well. So our conclusions here was that the sediments were not holding legacy herbicides, preventing the plant growth. We've actually sent the sediments out and had them analyzed also, and there were no residual herbicides from the ones that were treated over the last 10 years. So we're pretty comfortable that's not the reason. Of course, Hurricane Irma had gone directly over right at the same time the hydrilla was disappearing, stirring everything up. That could have implications, but we never, it's still research interest, but we know that it's not the legacy herbicides in the sediments. One of the other concerns we have over the time is that every year over 300,000 people move to Florida. With that huge population increase comes urban development, increased infrastructure, highways, sewage treatment centers, septic tanks that we have to be concerned about. So we were interested in looking at changes in the landscape, land use, and potential changes in our lakes. We also realize that we have these long-term cycles in rainfall. You're all aware we had fires and huge droughts, decadal droughts, and then now we're starting to get back in a wet cycle. So we want to look at rainfall impacts in addition to land use changes. So one of our graduate students, Chao Zhang and myself, put together these data, again, merged large databases. Water management districts have 5-year flyovers where they actually measure the entire district and look at land use. They digitize it so you can get at changes. One of the, we'll go through the conclusions on it in that lakes in areas of high agriculture or increasing agriculture tended to have higher nutrient concentrations, more so than background, suggesting that agricultural practices, fertilizers, potentially increased stormwater runoff from tilled grounds, and other aspects potentially increase nutrient concentrations in our lakes. So we have to be careful about that. A surprise to us was that in urban areas, high urban areas, or those areas increasing urbanization, didn't show any changes with nutrient concentrations in the lakes. That was not what we were expecting, and potentially this explanation could be the stormwater management within these systems is better. It's improving runoff, decreasing nutrients into the lakes. Also, as urban areas get bigger, they have central sewage instead of septic tanks, which is another hypothesis that we still need to look into. Cumulative rainfall deviations, we did see that over a 20-year period, there was strong correlations between cumulative rainfall, both positively and negatively, suggesting that individual mechanisms within lakes change based on the morphology and uniqueness of individual lakes. That's an ongoing research that needs to be continued, but now, thanks to all the volunteers collecting data, we have the data we need to address these. Another issue in the state, when I mentioned all the population increase, as people come, they need water. Florida is one of the unique states that gets 95% of their potable water from groundwater. And that is not sustainable. The more we take, the sooner the water goes down in those aquifers. Many of you have already heard that some of the coastal areas have used so much groundwater that they have turned salty because of intrusion from coastal areas. So we merged all the well data we could get from Florida Department of Environmental Protection and individual counties that measure water chemistry in superficial aquifers, and we matched that up with our lakes. And we found some very interesting correlations with pH, alkalinity, and specific conductance. So with this information, we're now able to look at lake regions and our phosphorus zones that we've developed with LakeWatch data, and they could be a good spatial framework to analyze shallow groundwater management. Again, another tool for us to be able to better manage both our lakes and the groundwater, which is very important for our continued growth in the state of Florida. Now we've been collaborating with some partners over from Denmark for quite a while. The last couple of years, they've come over here and stayed with us in our LakeWatch laboratory for 2 or 3 months at a time, where we compared our lakes from Denmark with our Florida stuff. They're both shallow systems with a lot of aquatic plants, and so we thought it'd be a very interesting comparison. One of the things we looked at, because they've had a lot of biovolumes for algal analysis and chlorophylls, as have we. And we wanted to compare and see what is best to measure, chlorophylls or biovolumes. Biovolumes take a long time because you actually have to put the sample underneath a microscope, analyze algae, and actually measure the sizes to determine the volume. Our conclusions at the base of it, all this work, was that chlorophyll, mostly because of the cost benefit, is recommended over biovolumes for monitoring long-term trends. They also provide better relationships with nutrients and SECIs that will allow us to predict changes down the road. Very interesting manuscript, and it's fun working with our Danish counterparts. Just to show you that LakeWatch data, we go from state to national to worldwide. Very valuable data. Another paper that we published with our Danish friends, we compared individual lakes with the same phosphorus concentration, and we looked at algae, aquatic plants, zooplankton, fish populations. And there's very significant differences in lakes, even though they have the same amount of nutrients in them. These we relate to the temperature differences, because the Danish lakes are much cooler. that have less of a growing season because of light regimes. And so with these models that we've presented, we're going to be able to make predictions of what the Danish lakes may look like as temperature changes over time. Very important, the Danish government is very interested in these models. It was fun working with them, and we will continue this in the future. Finally, another very interesting program. Again, I mentioned aquatic macrophytes. We measured a whole bunch for many years. Decreasing staff and budgets have kind of cut that back a little, but the data we had available with all of our water chemistry, I've shared with authors from 20 other countries where we've put together all this data from the world, and we're beginning to look at global determinants of macrophyte diversity patterns, why species occur where they occur, and developing good models, trying to understand the differences and potentially how invasions occur and that type of stuff. Very intriguing. It was fun working with all these worldwide researchers, and Lake Watch is right there amongst them. Again, showing the value of the amount of data that we collect and share with everybody who's interested. So summing it up, we do a whole lot with the data. We make sure everybody gets it. It's used by individuals. It's used by individual county governments. The state of Florida, the United States uses it, and the world. And as a volunteer, I think you should be extremely proud of your contribution both to lake science and lake management. We thank you. Continue sampling. I hope we get back to normal so we can visit face-to-face again, but thank you all for sampling. With that, I'll turn it over to your regional coordinators to finish up your meeting. Thanks for listening.